Let's look at another example. This is a very important example because you'll be hearing this throughout the rest of your career in the BMS department. Quick background. Bacteria such as E. coli, they need to eat sugar. Just like me and you need to get sugar in our cells, even bacteria need to get sugar. E. coli's favorite food in the whole wide world, it loves it. It's like gummy bears. E. coli's favorite food is glucose. It's a sugar. So glucose is a monosaccharide, and it's so yummy, and E. coli just loves it. But if E. coli runs out of glucose, like it eats all the glucose up, or there's no glucose in the environment, it'll go to its second choice, lactose. Lactose is kind of like the broccoli, and E. coli is like, ugh, well, I'll eat it if I have to. If there's nothing else around, I'll eat lactose. So lactose is actually a disaccharide. It's made up of two sugars, one glucose. So one of the sugars is glucose like this. The other sugar is galactose. And so you can see they're held together by this bond. So E. coli needs to make a special enzyme in order to break this bond. That enzyme is called beta-galactosidase. So beta-galactosidase is a special enzyme E. coli makes to break the bond in lactase and separate it into galactose and glucose. So when E. coli is eating glucose, its favorite food, it doesn't need to make any beta-galactosidase. But if it runs out of glucose, now it needs to make beta-galactosidase in order to digest the lactose. The gene needed to make beta-galactosidase is called LAC-Z. So write that down. LAC-Z is the gene coding for the beta-galactosidase enzyme. LAC-Z is in an operon called the Lac Operon. So remember, operons are genes that work together in a small area of the genome. So other genes that are in the Lac Operon include LAC-Y and LAC-A. But we're not going to talk about LAC-Y or LAC-A. We're going to just focus on LAC-Z. So this operon, upstream, you find the promoter, of course, right here in green. And downstream, you find the terminator, of course. Next to the promoter, as always, you have the operator. Remember, the operator will inhibit, if the repressor binds the operator, it will inhibit expression of these genes. And upstream of the promoter, we have something called the cap site. This is the activator region, which we'll talk about in a minute. Now, also upstream of the cap site, there's another gene called LAC-I. This is not in the Lac Operon. It's in an operon by itself, so it's kind of a lonely gene. And LAC-I has its own promoter, the LAC-I promoter. Now, if E. coli is growing in an environment with no lactose, so it's growing with glucose maybe, okay? So if E. coli is growing with its favorite food, glucose, and there's no lactose in the environment, then what's going to happen is LAC-I is going to get turned on. It's going to make this protein called the Lac repressor. So LAC-I is the gene that codes for the Lac repressor. When the Lac repressor is made, and there's, again, no lactose in the environment, the Lac repressor will bind to the Lac operator. So this repressor will bind the operator, which is upstream of the genes. And again, when the repressor is bound to the operator, this will block RNA polymerase and transcription is off. So normally when there's no lactose available, transcription of LAC-Z is off. Now, on the other hand, if there's lactose in the environment, what happens is the lactose, or a derivative that gets made of it called allolactose, these little triangle things, they're going to bind into these little nooks of the repressor protein. Now, the repressor cannot attach to the operator. It cannot bind because the lactose has changed the conformation of the repressor protein. So the repressor can't bind the operator. Now, RNA polymerase can come in and it can happily transcribe all of these genes. So we're going to make LAC-Z, and then we're eventually going to make the beta-galactosidase enzyme. Okay, so this is the operator binding to the repressor. No lactose present. The operator and repressor bind. Transcription turned off. Lactose is present. Then the operator and the repressor don't bind, and transcription is turned on. Now the Lac Operon is so complex and such an amazing piece of machinery. Not only do we have to think about the operator, we also have to think about the activator, this cap site. So here's how the activator works. Remember that the operator sensed lactose. So it sensed whether lactose was present or not. The activator, on the other hand, senses glucose. If there's glucose in the environment, then there's going to be low on this molecule called cyclic AMP. So in the presence of glucose, we either have low or basically no cyclic AMP produced. However, if the cell runs out of glucose, there's no glucose in the environment, then there's going to be a lot of this molecule called cyclic AMP. Cyclic AMP will bind to this cap site. So this is where the cyclic AMP binds. It binds the cap activator. If you have no glucose around, cyclic AMP will bind to the activator. And remember, this activates transcription. So RNA polymerase will come in and it'll undergo transcription of the Lac genes. So to summarize everything we talked about. E. coli prefers glucose. It's its #1 choice. If there's glucose in the environment, then there's going to be no cyclic AMP bound to the activator. So it's pictured in this one that has glucose and this one that has glucose. You see there's no cyclic AMP bound to the activator. However, if there's no glucose in the environment, then you are going to see, such as in this example, there's no glucose here, no glucose here, then you are going to see that cyclic AMP bound to the activator. However, even if cyclic AMP, like in this case, is bound to the activator, but if there's no lactose present, transcription still can't happen because that Lac repressor is going to block the RNA polymerase. So if there is no lactose in the environment, such as in the middle case here and the third one here, there's no lactose, the Lac repressor is going to bind to the operator. When the Lac repressor bound the operator, then the RNA polymerase can't get through and the operon's turned off. However, finally, if there is lactose in the environment, then the Lac repressor will fall off the Lac operator. So the Lac repressor is removed from the operator. And finally, the RNA polymerase can get through. So overall, in the presence of glucose, the operon is always going to be off because there's never going to be any cyclic AMP bound to the activator. In the absence of lactose, the operon is also always going to be off because the Lac repressor is always going to be bound to the operator. The only way the operon is ever going to be turned on is if there's no glucose, meaning the activator is bound, and if there is lactose, so that the Lac repressor can fall off, and now the RNA polymerase can finally get through and transcribe the genes. As always, I found the best YouTube video I could explaining all of this, and now's a good time to go watch it.